Have you ever watched a Drishyam movie? Suppose you are an investigating officer on that case. There was no witness, no evidence, no fingerprint, but one biological evidence that could solve the case. Now tell me what could be that biological evidence? Hairs, nails, blood samples? If this was your answer, tell me what does this biological evidence contains that could identify a criminal or identify a person? The answer is DNA. So, hello everyone, my name is Ashutosh Kosariya and today we will learn about DNA. We will mainly focus on the structure of the DNA, but we will also cover its function, where its location and how it is present in the nucleus. So, let's start with the definition. DNA is the hereditary material of a living organism. It stores and transmits genetic information from one generation to the next. So does DNA is like a bag where information are just stored, and information are stored in what form? Is it in the form of the binary language, 01 language, or it is like a paragraph, where father cells write a paragraph like make my son's height like this, make my daughter's ears like this, and send it to the next generation? No, no. The informations are present in the form of the nucleotide sequence. You will get a clear about this topic once we have completed this lesson. Okay. So if you understand this definition, try to answer by this simple question. So my question is, DNA is only hereditary material found in all living organisms. And second question is, DNA is the only genetic material of hereditary material found in all biological entities. Try to answer these two questions. Confusing, right? Let me clear it. There is only difference in these two sentences. In one sentence, I have mentioned there is a living organism mentioned here, and in the second question, there is a biological entities mentioned here. So let's clear your confusion. Just remember these points. These are a very basic points, but asked frequently in the many examinations in the form of the MCQ or SSR region type questions. So, biological entities, suppose this is biological entities, can be of two types. One is a cellular and one is a non-cellular. These fellows can perform their reproduction by this own, synthesize their own proteins. That is why they are known as living organisms. Whereas they require specific host for their reproduction and protein synthesis. That is why they are not considered as a living. They are non-living. So here comes all the bacteria or archaeas, protistas, fungi, plantae, and animalias. All of these are considered under living organisms, and everyone contains only and only DNA as a genetic material. Whereas here, non-cellular or non-living, here comes a viruses. They can contain DNA and RNA as a genetic material. They contain DNA or RNA, not both. So in my first sentence or first question, I have mentioned that DNA is the only genetic material found in all living organisms. So it is true, right? It is true because in living organism, only DNA is found as a genetic material. Whereas in my second question, I have asked DNA is the only hereditary material found in all biological entities. So here I have asked, in biological entities, DNA is the only genetic material. The answer is no, that is false, because in the biological entities, here comes a viruses that also contains RNA as a genetic material. Okay. So the second statement or second question was our incorrect. So let me summarize it. There can be a two type of nucleic acid. One is a DNA and RNA. Both can have that is a genetic material. RNA is genetic material only in case of the viruses, whereas other living organism only contains DNA as a genetic material. Got it? So let's move on to the structure of the DNA. I know most of you have aware about the structure of the DNA. It is look like this. It is a double-stranded helical structure having a hydrogen bond between the strands. Right? But I want you to start it from the basics. And from basics, we should consider few parameters. So let's understand these parameters first. The first parameter, DNA is polymer of nucleotide types. Second, they are double-stranded. Third, they are anti-parallel in nature. The two strands are anti-parallel. Anti-parallel strands. Fourth, there is a complementary hydrogen bonding between the nitrogenous bases, between the two strands. There is a complementary base pairing between two strands. And last but not the least, DNA is a double helical. Double helical in nature. Now let's try to draw the structure of DNA using this parameter first. Okay. Let me clear a space for that. So our first statement says DNA is a polymer of nucleotide, which means there is some molecule known as a nucleotide that is a monomer. This is a nucleotide. That is a monomer where the DNA polymer are formed. So suppose this is the one nucleotide. And similarly, many such monomer combine together to form a polymer. So here we are drawing our first statement. DNA is a polymer of nucleotide. Okay. So this is how DNA is a long polymer chain of a nucleotide. Second statement says it is a double-stranded, which means this structure is a double-stranded form. So I have made it double-stranded form. Third statement says they are anti-parallel in nature. To represent the anti-parallel in the strand, I am just hypothetically drawing a triangle here. There is no such triangle in the structure of DNA. I am just making it hypothetically to make you understand that there is something in the DNA strand that makes them an anti-parallel. Okay. So these two arrows represent the anti-parallel between the strands. And the fourth statement says there is a complementary base pairing between the two strands. So there will be base pairing between the two strands. And we are already aware that these complementary base pairing are nothing but hydrogen bonds. And last statement says DNA is a double helical. So if we rotate this DNA in a double helical form like this is the one polymer or polynucleotide chain, this is the second polynucleotide chain, and there will be hydrogen bonding between the bases. And this triangle here represents the anti-parallel nature of the DNA. Now you understand how this structure forms like this. If you understand this definition, or if you understand these points, answer my simple question. What is the full form of DNA? Full form of DNA. I think most of you have known that DNA is nothing but deoxyribonucleic acid. My second question. What is the deoxyribo represents here, and why it is called a nucleic acid? Third question. What is present in the DNA that makes them anti-parallel? And what do you mean by the complementary base pairing? You will get answer to all of these questions once you have understand the individual structure of this monomer nucleotide. So now let's try to understand the structure of the nucleotide. So this is how an individual structure of nucleotides looks like. There is this molecule represents a 5-carbon sugar. This is a sugar, which is a 5-carbon, and this known as ribose sugar. Okay, and here is his 5th carbon. Okay, so this represents the sugar. And this molecule represents a nitrogenous base. This is a 9-membered ring of nitrogen and carbon. This molecule represents nitrogenous base. This is nothing but a nitrogenous base. And this molecule, this one, is a phosphate group. Phosphate group. Understand? First prime carbon, or carbon number 1 of the ribose sugar, and the ninth number nitrogen of the nitrogenous base forms a glycosidic bond. So carbon number 1 of sugar and N9 or N1 of nitrogenous base forms a glycosidic bond, and this together, sugar plus nitrogenous base, are known as nucleoside. Got it? And this phosphate forms a phosphoester bond. Got it? And all this structure, sugar, nitrogenous base plus phosphate, collectively known as nucleotide. Okay, so phosphate, sugar plus nitrogenous base known as nucleotide, and sugar plus nitrogenous base only known as nucleoside. Got it? And now I am answering your question, why it is called the deoxyribose. Okay, so if you see the second position of the ribose sugar, here the oxygen is absent from the second position. That is why it is called as deoxyribo. The deoxy term because there is oxygen is missing. Ribose comes from because this is ribose sugar. Okay, deoxyribonucleic acid. And acid what comes because of this phosphate group, because it contains the hydrogen. It can donate its hydrogen, and one can which donates its hydrogen are known as acid. That is why DNA is known as acids. It is acid. Now we will learn about different types of the nitrogenous base. Nitrogenous base. There are 2 types of nitrogenous base present in the nature. One is a purine, which is given in PU, It is a purine. And second is a PY, known as pyrimidines. And these purines can be of two types, adenine, guanine. And these pyrimidines can be of three types, cytosine, uridine, and thymine. Got it? There is a rule that purines always base pair with the pyrimidines in throughout the strand. So it is the purine and pyrimidines where the hydrogen bond take place. And there is a rule of complementarity that A, which is the purine, pairs with a double hydrogen bond with T. And reverse is also true. Okay? T forms a double bond with A. So this is the one rule of a complementary base pairing. Now understand what is the complementary here. Similarly, C forms a triple hydrogen bond with G, and G forms a triple hydrogen bond with C. This is the second complementary rule. Got it? This point is to be noted here that this uridine is only present in the RNA, and T is absent in RNA. This is a very important point. Okay? And U forms double bond with A. A forms double bond with U. This is the third rule of complementarity. Got it? Now let's see the individual structure of this nucleotide bases or nitrogenous bases. This is how adenine looks like. There is a amino groups in the sixth carbon. So it is also known as 6 amino purines. And there is a amino group in the second position and oxo group in the sixth position. That is why they are known as 2 amino 6 oxo purines. Similarly, there is a cytosine, uracil, and thymine. Thymine is nothing but a methylated uracil. Okay. If you observe this two structure carefully, there is the only difference is the presence of this CH3 group in the fifth number of the carbon. So this is the difference between thymine and uracil. Now let's understand how this monomer of nucleotide forms polymer or DNA. I have to remove this oxygen because we are making DNA here. One more thing. The oxygen in the 2' portion of the ribose is present in case of the RNA and it is only absent in case of the DNA. And that is one difference between the DNA and RNA. Okay? So if you are making the RNA, you do not have to remove this oxygen atom. So let's see how this individual nucleotide molecule forms a long polymer. So this is the first nucleotide. This is the second nucleotide. What happens here? The phosphate group attack the nucleophilic attack on the OH groups present on the 3' of the carbon of the ribose sugar. And because of that, there is a formation of bonds, ester bond. So this is the one ester bond, one phosphoester bond. There is a second phosphoester bond, and together they are known as phosphodiester bond. Got it? Similarly, many such nucleotides are attached with the phosphodiester bond with one another and forms a long chain of a nucleotide. And that is how a long chain, it is a one nucleotide, this is the another nucleotide, this is the another nucleotide, this is the another nucleotide. All are connected with the phosphodiester bond. Got it? Similarly, here also There are one nucleotide, there is another nucleotide, there is another nucleotide, there is another nucleotide, all are connected via phosphodiester bond. And one more thing, these two strands are connected with the hydrogen bond between the nitrogenous bases. As I told you earlier, that there will be two hydrogen bond between the A and T. Here you can see there is a two hydrogen bond. And there is a three hydrogen bond between the C and G. So here represents three hydrogen bond between C and G. Okay. And one more thing, I have said purines always pair with pyrimidines, and pyrimidine always pair with the purines. So if this is a purine, it should be a pyrimidine. So yeah, it is pyrimidine. Suppose this is a pyrimidine, the opposite strand must be, or complementation must be purine. So this is a purine. Got it? And one more thing, suppose this is the last nucleotide. This represents one last nucleotide. If you observe here, you will notice that there is no phosphodiester bond possible this side. So this group is free. So our 5' carbon is free. Whereas in this end, there could not be possible any further phosphodiester bond here. So this 3' end, this 3' carbon OH is also free. That is why these ends are known as the 5' end and these ends are known as 3' end. If you see it in the another stand, if you see it in this stand, it is just reversed. Here, the 3' piece is free and in this side, 5' end is free. So if you observe it carefully, these both stands are anti-parallel orientation. Both are parallel. Both are parallel. But they are in opposite orientation. That is why they are known as anti-parallel in nature. Got it? So this is how the structure of the DNA looks like when we combine all the monomers. So this is how this represents a backbone of the DNA. If you observe here, these backbones are made up of phosphate and sugar moiety. Let me move back to one slide and show it one interesting thing here. Observe this carefully. If you observe it here, this is nothing but a sugar and phosphate. What does it represent? It represents the backbone. Backbone of DNA. And there is a charge in the phosphate. That is why DNA is negatively charged because of this phosphate. Okay. One thing I forgot to explain in this slide. Suppose I am saying my DNA is made up of 5 base pairs of nucleotide. 5 base pairs of nucleotide. So what do you understand by this base pair? Does this is the one base pair? Does this is the one base pair? No. Base pairs are the complementary nitrogenous base between the two strands attached with the hydrogen bonds. So these two represents a one base pair. Similarly, in this diagram, these two represents a one base pair. Got it? So can you tell me how many base pairs does this diagram contains? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So total 12 base pairs of nucleotides this DNA strand contains. What? So now you tell me how many nucleotide base pairs humans have or diploid human cells have. So we have approximately 6.4 into 10 to the power 9 base pairs of nucleotides. I am talking about diploid cell where two sets of chromosomes are present. So if you observe this diagram, this gap is evenly maintained throughout the length. And this is true in case of the human DNA, that This gap is around 0.34 nanometer in length. So suppose this is the one DNA strand, this is another DNA strand, and this gap is evenly maintained throughout the length of the DNA. That is the 0.34 nanometer. If we write it in a nanometer, it would be around 0.34 into 10 to the power minus 9 meter. Got it? Now we have the length between the two nucleotides. We have total number of nucleotides. Can we calculate the exact length of the human DNA? Yes. If we multiply this number by this number into 6.4 into 10 to the power 9 base pair. So this 9 get cancelled and it would be around 2.17 something meters. Got it? So this is the length of the human DNA. Understand? Human DNA is approximately 2 meter in length. So how does such a long 2 meter DNA can fit into a tiny nucleus? The answer is packaging of the DNA. So that is how DNA is packed inside a chromosome. Or chromosome is nothing but a complex of the protein and DNA. So what happens? It's quite clear with this next diagram. Here DNA, this is a DNA. It forms a complex, or DNA is wrapped within this protein known as a histone protein. So DNA firstly wrap around this protein, then entire this protein DNA complex, it is known as a nucleosome complex, it wrap around itself and ultimately it forms a chromosomes. This is how chromosome looks like. Alright. So that is how a chromosome looks like. It is nothing but a complex of a protein and DNA. This is a very huge topic known as packaging of the chromosome or packaging of the DNA. We will understand in the next class. So does this 2 meters of human DNA, 2 meters of human DNA is fit under a single chromosome? No. This 2 meters length come from the combining all the chromosomes DNA. So how many chromosomes do we humans have? We have 23 pairs of the chromosomes or 46 chromosomes. So collectively, if we combine the length of this entire chromosomes, then we will get this 2 meter length of the DNA. Alright. So can you tell me how many DNA we have? In this diagram, this one DNA is packed inside the chromosome. What does it mean? Which means one chromosome represents a one DNA molecule. So if we have 46 chromosomes, we have 46 DNA molecules. Got it? So this is the karyotyping of a human chromosome, representing all the 23 chromosomes. So from here to here represents the autosomes. Autosomes, and this known as 23rd chromosomes or sex chromosome. If this is present inside the human body, human cells, it would be a male chromosome. And if this is present, it would be a female chromosome. Got it? And one thing, chromosome number 1, it is the longest chromosome. And chromosome number 21st is the smallest chromosome. So can we say that if chromosome number 1 is a longest chromosome, it contains the more number of DNA or more length of the DNA? Yes, it is also true that if I am saying that chromosome number 1 is the longest one, it contains the large amount of the nucleotides. It would be around 250 base pairs of the nucleotides. Whereas in case of the 21st chromosome, approximately 50 base pairs of nucleotides are present. So this difference in the length of the chromosome is because of the difference in the size of or number of the nucleotides. Got it? And these are present in the form of the duplet. Every chromosome are present in the form of duplet. Why? Because we organisms are diploid. Suppose if we are triploid, then it would be present like this. There will be another copy of chromosome present of entire this chromosome set. Got it? And suppose if we are saying some cells of our body are haploid, and in that case, if we observe their chromosome, it would be like this. So that is all for today's class. Hope you have understand whatever I have taught you right now. Video has already become very long, so I have saved some topics for the next class. But before leaving the class, I want to answer the question I have raised in my first slide, that how a DNA can be used to identify a person or criminal. For that, you have to imagine a scenario. Suppose this is a chromosome. And I am extracting a DNA from here. I'm extracting a DNA. And we know that DNA is nothing but a sequence of nucleotides. A, T, G, C, T, A, T, C, G. Similarly, they're complementary. T, A, C, G, A, T, G, C, and so on. So DNA is made up of this. So DNA is nothing but this sequence only. So suppose this is my DNA sequence of one chromosome. Okay. And this is, suppose this is a your DNA. What happens in the DNA? We humans share 99.9% of our DNA. So if this is our DNA, mine is yours, the 99% of this DNA are approximately similar between my DNA and your DNA. But this 1% DNA is different between the individuals, any two individuals. And this different DNA are known as marker region. There are lots of marker regions are there, like VNT area, SNP regions are there, and many more. So these regions are the one that makes a different individuals, mixed or used to identify individuals. So if we extract a DNA sequence from this region and try to match with this trend, this is your, this is mine, definitely it will not match. So similarly happens in the identification process. Suppose this is the crime scene. We got a sample from there. We extract the DNA. If DNA is in a very small amount, we have to amplify it or multiply it so that we get a huge amount of DNA. Then sequencing is performed. Similarly, suspect DNA is extracted from their respective biological samples. Their amplification are done, then sequencing are done. Then these two sequences are done, sequence matching. So if this suspect is present on this crime scene, definitely that biological evidence would be of his only, and that sequence will match 100%. So that is how DNA sequencing can be used to differentiate a person or identify a person. But it requires very expert and sophisticated instrumentation. That is why generally they are not used in the forensic science. A DNA fingerprinting is used. If you are interested to learn about this DNA fingerprinting, I can include that how it can be used to identify a person, how it can be used for the paternity testing. So if you're interested in, I can include that in my next class. So that is for the today's guys. Thank you.